Окей, как вас зовут, извините? Даня? А, Даниил, да? Окей, хорошо. Yeah, пожалуйста. Yeah. Okay, so hello everyone. Hello, Daniil. So we welcome you to the Art and Science Master's Program Open Day at Ichmon University, and today this Open Day will be devoted to Bioart, one of the majors of our program. My name is Alia. I'm the head of the center. Laura Rodriguez Torres is curator of Bioart major in the program, and Victoria Romanova is our graduate and also a member of the team, of the AR team, coordinator at our gallery. So let me tell you a couple of words about the master's program. Sorry. So it's an international master's program. Я могу вообще без них. Ну там у нас не будет. Потише? Она хорошо слышно в зуме. Окей, хорошо. So this is an international master's program which is taught in English, and it's the program that gives you opportunities to combine artistic research, artistic practices, and create your own projects on the intersection of art and science and technology. This year we have three majors. One of them is digital art, the other one is technological art, and the third one is bioart. And today Laura will be presenting bioart major. So the first thing I want to start with is who is this program for? Who can apply to the program? Basically, students with any academic background. If, for example, you are artists who are interested in kind of mastering some technical things, mastering some technology so that you can create your projects, you can apply to the program. If you are scientists or researchers or engineers who want to find a new way of looking at your projects, who want to bring artistic practices to your research work, and again to create your own projects, you are welcome to the program. If you are curators, art historians, so basically graduates of humanities majors, you can also apply to this program because you can develop your curatorial projects within this program. So basically, it doesn't matter what you graduated, what program you studied before, you can apply to Art and Science Master's Program. And head of this program is Dmitry Ozerkov. He's head of the Department of Contemporary Art at the Hermitage Museum. He's the director of Hermitage 2021 project, art historian, curator. And since I told you that this year we have three majors, we have curator for each major. So Laura is curating bioart, Vadim Smaktin is curating digital art, and Maria Kovtsova is curating technological art. Well, this is basic information about the curriculum, but I wouldn't focus on that, but would rather tell you about the benefits of the program and what this program will give to you as the students of this master's. Well, first of all, you will work with professional artists, scientists, and curators, such as, well, as I've said, Dmitry Ozerkov. Just a moment, I'm going to switch to the slide with the speakers. So Elena Nikonova, with basically all the best art and science interdisciplinary artists in Russia, and not only in Russia, are speakers of the program. For example, Natalia Fedorova, she's a curator and researcher working with media art. Helena Nikonova is one of the basically rare artists in Russia who works with AI, neural networks. Dmitry Morozov, I think everyone knows Tom, he's working with robotics, mechatronics, and sound. Ippolit Markelov is working with bioart, and Daria Kubov, digital artist. Ekaterina Nikitina, she's a post-humanism expert. Boris Shishnikov is working with sound. Daria Milya is a curator from ZKM, one of the key institutions in the field of art and science. Then we have Robertina Sibianic, who's artist who's based in Slovenia, also internationally awarded, and she's received honorary mention and at Ars Electronica Festival. Lindsay Walsh, also the graduate of Symbiotica Lab. Maya Smrekar also received honorary mention at Ars Electronica, and Dmitry Zarkov I mentioned as well. So working with these people in different formats, you will have an opportunity to create your own project, to get their feedback. And this year, for example, we introduced such program as Artistic Studio, not a program, but such course as Artistic Studio. So we work in artistic studios with Елена Никанолия, with Vtol, with Ippolit or Ildar, and can receive their feedback straight away. Another benefit, so basically, as a result of this, you become the part of this art and science community in Russia and internationally, and can create your own projects while you're studying, basically, and present them here in St. Petersburg or internationally. For example, we opened this gallery in September, and starting from October till January, students had 4 exhibitions. One of them was on sound art, the other one was held within the curatorial forum in November, the other one was in January, it was on bioart. So you have lots of different opportunities for showcasing your projects. This is the photo from the exhibition of graduate projects. You can also develop your curatorial projects. So a group of our students created this grounded project, which is in fact an international project. They made a fantastic educational program together with the Soil Museum in St. Petersburg, and they are preparing an exhibition at the moment. Well, basically, you can present your projects internationally, because last year, 14, I counted students who participated in the project, 14 students participated in the exhibition in Ars Electronica Festival. So I think it's a fantastic opportunity, in fact, for anyone who is a student in this field. And 6 students, first-year students, participated in the public program with their workshops. So again, we're striving to give all the best opportunities to our students. So you become the member of this community, you can present your projects, and of course you study in English, you can communicate with different speakers worldwide, you can travel for exchange programs. Well, we hope that you'll be able to do that once the borders are open. And yes, I told you about the speakers, and now I want to give the floor to Laura, who will tell you about bioart and the course itself. Yeah, Hugo.
Okay, as we have said, we have this year, we will open these 3 specializations. Although we create a program where all the students will collaborate and will learn from the 3 of them. For example, we have some collaborative workshops at the end of the semester, in the 2nd semester and 3rd semester. So although you can choose that your major will be with this specialization, you still will be able to collaborate with digital art and with technological art. My name is Laura Rodriguez. I'm a graduate from the master's program, but I also have a master's degree in molecular biology from Skoltech in Moscow, and previously of that I studied biotechnology. So I have 8 years of experience in science, but around 3 years ago I decided to change. I guess that my motivation was that I always wanted to create something. I have this sparkle that I wanted to create art and also share the knowledge of technology through it. So in the bioart specialization, we have put a last name of it is Biomedia and the New Nature, because we will be dealing with a lot of the organisms. We will be creating with them, collaborating with them, and also imagine a lot of the futuristic scenarios of a new or another nature. So when I say, or people ask me, what do you do, what is bioart, they mostly think that I'm painting with bacteria, like you see in the image. I have never painted with bacteria, so this is like a very common misunderstanding. This won't be art or bioart, this will be like a craft. Do it with biomedia, with bacteria, or with biotechnology. Don't think that this is not easy. Usually you think that, oh yes, I can play with bacteria. Your first attempt will look something more like this. However, I repeat, this is something that will be a craft with bioart. When we are talking about a craft with biotechnology, when we talk about bioart, we are talking about a branch of contemporary art. So something that is very important is the conceptualization behind. And bioart is actually something not that new. I think it had the boom or the hype in the 80s. For example, Eduardo Katz, he created this is one of the most famous bioart projects, and he coined this term of bioart. You can also know this tendency as biological arts, transmedia, living art. And what you're seeing here, this is Alba, a bunny that was modified genetically. They took a gene from the jellyfish that expresses the protein that fluoresces under blue and UV light, and inserted in the genome of this little bunny. This is something quite common in science. Before we are using these indicators of the proteins to detect some tumors in mice or in different organisms. But this was the first organism created for the sake of art. Why is so important? Well, I need to say that there is a lot of speculation if the rabbit really existed and glowed like this, because we only have this picture. I don't think that it really matters if it existed or it glowed exactly like this, because the artist already put it in our mind. He already put a new organism in our mind, that the possibility to create something of a new nature. And that's why it becomes so important in the history of bioart, and also because we're talking now that this is not just an object of study, but it's a subject. And now we're thinking of the ethical issues and the level of importance of the other organisms comparison with humans, because we are so used to it in this whole Anthropocene or the era that we think that everyone was around humans. Another more updated example is from Joe Davis. Joe Davis is an American artist that he developed 3 years ago an artwork in a workshop where what you see here in this blast is silk made of gold. So basically it's a fairy tale from Art&Science coming to life. When you can, something kind of alchemy. So basically they took the genes from a marine sponge. This protein that comes from the gene is capable to catch different metals or materials, and inserted the gene in the silkworm. So the silk produced by the cocoon had this protein. When you put the silk inside a solution with gold, it will catch all the gold, and you will have this silk made of gold, basically. One of the approaches not only to present your artwork, but it's also to make workshops around the world. So we had the opportunity to attend this workshop in Moscow and learn about how they make the genetic modification, how they collaborate with the scientists in Harvard, where he is based right now. And then let's go a little bit about the history. So as I told you, it's something not as new as a lot of people think. So in 1980s, another artwork of Joe Davis is called Micro Venus. So this is the first of the artistic works that construct a synthetic DNA. So they took an image. This is the image of the ancient rune that represents Earth and humanity, translated into series of ones. That will be the binary code. You can see still here the image. And then assigned to each of the 1 and zeros a different codon of the DNA. So they create a synthetic DNA, and basically they send it to the space. And why? Or why not? But basically it's because this artwork was an answer to a previous scientific project, not artistic, but scientific, developed by NASA and Carl Sagan, where they sent in the spacecraft Pioneer 10, they sent this Pioneer table. Basically, in case this spacecraft finds some other life, and this other life or entities will understand where this spacecraft comes from or who we are. So that's why we have a diagram of male and female. Here you can find our position in the solar system, what are the atoms that we are made of, or they exist with major frequency in the Earth. However, you can see the male figure that is quite accurate representative, especially you can see the difference in the genitalia of the two of humans. So they noticed that there was not such accurate representation of the female figure. That's why Joe Davis decided to send years later this, like, you know, we forgot to send this part. So this is also kind of the idea behind the artwork. What is the concept? Like why art is usually a fragile entity and it's always reacting at what is happening around. So that's why also in the 90s it becomes so popular to work with bioart. And one of the first hubs of bioart was created by Oro Katz and Yemen Sur. So let me ask you, what figure do you see here? Yeah, right, it looks like a coat, like a dress. So precisely this is called victimless leather, or victimless jacket, someone also call it. So they took cells from a pig and cultured it in a shape that looks like a jacket or like a dress. So basically they are talking about the ethical issues of using animals for human benefit, for our clothes, or to obtention of leather or food. And the idea of grow our own clothes without killing animals. This artwork, as amazing that it also become more popular because these dresses started to grow and grow and grow during the exhibition. And it becomes something alive that the visitors will see how it was growing from one week to another. And when the exhibition ended, the main question is it alive? Can we kill it? If we disconnect it, what is the ethical issues? Also working with cells, not only with organisms. Another idea of organisms that we can work in biological art. So I mentioned DNA, I mentioned cells. So now let's go to some of the higher organisms like plants. We have the Institute for Inconspicuous Languages, reading leaves, from Stella Petric. And basically what she was doing in the plants, there are small stomas. These stomas are the openings of the leaves that catch the oxygen. They are microscopic. But when you see it in the microscope, they look like leaves. So they are little holes that are moving, opening and closing all the time, taking air and taking CO2 and put it back oxygen. So she made collaborations with different specialists that they know how to read leaves. And then they designed a language where they, by a neural network, that it was reading these stomas as if they were leaves. And then they started to understand what these stomas wanted to say. Can we put back the video? So in the installation, they have all the flowers and the plants where they were analyzing, and the observer could also see through a microscope the different stomas moving. And then we have here some of the words. Of course, some of them doesn't make sense, and I think this is very accurate because it doesn't mean that plants should make sense for humans. We're just trying to understand how there's an alternative language between them, maybe. Okay. Then we have also collaboration between plants, bacteria, and technology. This is an artwork from Gilberto Suárez. He's a Mexican artist dealing with the topics of ecology. He is developing a lot of different robotic entities, trying to decrease the environmental effect that humans have made in the cities. For example, he created this installation. Basically, in the center, we have a bioreactor with different algae that are connected to a bacterial cells from the soil of Mexico City. And when we put water from the waste of the city, this entity becomes alive. The bacteria in the bacterial cells receives the oxygen and water treated from the bioreactor in the center, and it can generate electricity. This electricity is transformed into sound and flashes, and the light is necessary so the algae and plants can make photosynthesis. So it's a loop of surveillance by making the residual waste of the city potable or drinkable again. Then if we go to the next artwork, when in the beginning of the presentation I told you that painting with bacteria might be something considered just as a craft. However, the artist Melissa Fisher, she's also taking this growing of the bacteria as a technology, but she's making sculptures of her own body, taking the bacteria that lives in her skin and culturing in these sculptures. The sculptures are made of agar. There is nutrient media with sugars and different proteins, so the fungi and the bacteria can thrive in these sculptures. In this case, she's talking about the identity. So now in your body, you have hundreds of bacteria in your skin and thousands inside your gut. And all of them are making their own biochemistry. The subproducts here are also interacting with your own biochemistry. The smell that you have that is different to another person is also because of the bacteria that lives in you. So she's making the question, who am I? What are all these other entities that live in me and make me? Okay? Where I should point? Here. Okay. And going back to the modification of organisms, Eduardo Katz also created a modified petunia. This petunia had a gene from the artist, so they analyzed the genome of Eduardo and took the gene that codes for the hemoglobin in his blood and inserted it in the petunia. Now the name of this petunia is Edunia, a combination between Eduardo and petunia. And you can see the veins of this flower that possess the blood of the artist. This is again talking about the subjectivity of the plants, the implications of collaboration between humans and plants. And a lot of people was amazed and surprised of, for example, Alba the rabbit or Edunia, but some others also think that it was something like a monster. For example, the glowing rabbit, that is something not so natural. What I like in one of the conferences that Eduardo Katz made in this moment when he's talking that people was afraid of these organisms, he asked the public to put their hands a little bit together, not completely like touching around 5 millimeters separated, and you will start to feel some heat. It's normal because we have some temperature in our body, but part of this heat is also infrared light. Our eyes cannot see the infrared light, but there are other organisms like reptiles or bats that they can see the light. So people was afraid of the glowing monster of Alba, but some of us, well, all of us look also like a glowing monster for some other species. So this is the idea of changing our perspective, or human or anthropocentric perspective. That is something that BioArt deals a lot. For me, when I hear this in this conference, it was really funny to understand that I'm glowing all the time. So even when there's a dark climate or gloomy weather here in St. Petersburg, I can say, okay, I'm glowing. I'm okay. So I explained a little bit of the artworks, the most famous of the history of science art, a few. Now I want to go to the students' work that we are doing, and for that I want to introduce one of the speakers that Alia mentioned. Lindsay Walsh is an American artist. She is now based in Berlin and is delivering a course for us that is based in the Biomedиа, working with different cells from humans, and from plants, and she has a message for everyone that is
Hello,
here.
future ITMA Art and Science students. I hope you are excited for the wonderful road ahead of you, exploring the interconnectedness between art, science, society, and culture. I'm American artist, designer, writer, and researcher Lindsay Walsh, and I teach the course Art, Natural Sciences, Aesthetics, Theories, and Discourses. In my course, we cover all the practical applications. Hello, future ITMA Art and Science students. I hope you are excited for the wonderful road ahead of you, exploring the interconnectedness between art, science, society, and culture. I'm American artist, designer, writer, and researcher Lindsay Walsh, and I teach the course Art, Natural Sciences, Aesthetics, Theories, and Discourses. In my course, we cover all the practical applications to working with living materials, ranging from the microbiome to tissue culture and more. With the emergence of new technologies and scientific advancements, the world is rapidly changing, and I look forward to seeing all the things that you're going to do with your transdisciplinary education at ITMO University.
I'm not sure if you've heard everything. Not really. Can we solve this? Получается? Погромче поставить, ничего не слышно.
Hello, future ITMA Art and Science students. I hope you are excited for the wonderful road ahead of you, exploring the interconnectedness between art, science, society, and culture. I'm American artist, designer, writer, and researcher Lindsay Walsh, and I teach the course Art, Natural Sciences, Aesthetics, Theories, and Discourses. In my course, we cover all the practical applications to working with living materials, ranging from the microbiome to tissue culture and more. With the emergence of new technologies and scientific advancements, the world is rapidly changing, and I look forward to seeing all the things that you're going to do with your transdisciplinary education at ITMO University.
Oh, Laura, if you can say a couple of words, because she's working with living, with tissues and tissue culture.
So yeah, during this course we are analyzing the microbiome, the organisms that live in your body. We're also working with tissue culturing, creating mini sculptures with different cells from mice or pigs. Also learning how to remove these cells. And she is talking a lot about the queer biology. Also one of the philosophies that she introduced in the course is the systems of care. How do we can improve our empathy towards other species? So I think better to explain what we were doing, I will show some examples that we had in the Bioart showcase last January. So I was being the assistant of Lindsay during the course, and I also get inspired of the techniques that she was teaching us. So she, for example, taught us how to remove all the cells from a spinach leaf. And then we understood that we could also try other different plants. So I started to make experiments of it until I, for example, got the whole flower almost without any cell. And this inspired me to think about the shape of the organism, because here people still see a flower, but there is no plant anymore. It's just a shape. There is a cellulose structure, and in the laboratory we're able to seed back cells into this structure. For example, for mice. And if there is mammal cells that could be seeded back here, well, that makes the idea that we could also seed back or seed new cells from human in these structures. The scientific research behind of this, or how this technology evolved, is that they are trying to take this cellulose structure to make a transplant of myocard tissue, because the leaves of the plants have already a lot of veins. So for example, when there is a stroke in the heart and a little part of the tissue stops moving and there is some necrosis, there is the speculation that it could be possible to use another type of tissue, seed it back with the myocard cells and make it functional. So I decided to make a speculative artwork where we can think about transplants, aesthetic transplants of different leaves. And that's why the name it evolved to becoming plant, because we're talking about the malleability of the shape, and if we remove all the cells of the plant, what means to be a plant? So also, what means to be human? Are we just a bunch of cells working all together? Can we try to understand the essence of what is a plant or what is a human through modifying our shape? And our students also create different artworks, Natalia Molina, she was working with the same technology by removing the cells of a frutal sculpture, and she made these little mice. And the evolution of this project is that she wants to put here layers, a layer of cells of the epidermis of a mice. So around the 70s, the scientists took cells from a specific mice, and now these cells are all over the world, are our model organisms for cells. And everyone has in the laboratories the cells called C12B12. That's the scientific name of this strain of cells. And we use it just as an object of study. So if someone wants to make studies with the skin, they probably will use these epidermal cells of the mice. So she was thinking about how to give back the essence of these mice, how to recognize again that it was a mice, it was an organism, that it died 70 years ago, around 50 years ago in the 70s. No, 50 years ago. 50 years ago in the 70s. But we still use its cells, so it's some part of it is still alive. And also thinking about how we treat animals inside the laboratories. Okay? And moreover, she's talking about something that can be so traumatic, like the use of these organisms in science, but the installation also is making this beautiful contrast of the pink. The pink color is because is the color of the medium that we use to culture the cells, so they get the nutrients from this medium. And also we have different collaborations inside ITRO University. I think one of our advantages is that we are inside a technological institute. So we have different labs from robotics to nanomaterials and also biological. So there is an ongoing research in the nanomaterials laboratory where they are injecting ferromagnetic nanoparticles into spiders. These nanoparticles react to magnets. So when you inject it into the spiders, the silk that they produce contains these nanoparticles. So it reacts to the magnets. And the students, it was a group project of all the students that took the course, they were thinking how to give a technology to the species, how the spiders could use this technology for their own benefit, or if it is ethical to use only this material for human benefit. That's why the name is Living Labor. And they create this structure, is this sphere, with all the magnetic silk. I hope the video will allow us to see how it was moving, because inside of the sphere there was magnets moving, and every time there was a movement of the magnets, the silk was moving. A lot of people thought there was something alive moving there. It really looks like an organism breathing time to time. So this is one of the final projects that they did in less than 2 months, learning the technology. So it was a very intense course, and we try that every course that you have, you will have an outcome. So it will increase your ability to create some projects. So now I would like to give the stage to one of our current students, and Jenny, please, he will also share with us what is he doing in the area of bioart.
Hello, my name is Dень, and I'm a multimedia artist for 3 years, and now I'm a first year student of Art&Science Master Program, and I would like to tell you about our project Plants Vision. Ethan, is it okay with my call? So this is our, how we use it? Yeah. So this is our team. We have 5 of us to manage with all this very complicated project. So the idea of this project is to create some communicative system to interact with us as humans, and we used to think that plants is like some sort of decoration, and how we can make our apartments more beauty, but they is what about our research. Here you can see a photo, and my question for you: how do you think what is it? How it looks like? So here you can see a portrait.
Like a human,
So,
yeah,
it's
face.
a human. So and this structure is cells. All this round circuits, it's cells of leaves. And we have found experiment which was produced in 1906 and described in Oxford Journal. And here you can see another photos which made through lenses, through cells which act like lenses, and these layers. I will show it where it is like. Oh yeah, here. So here is a leaf, and this is ocelli. So we can see here. So it acts like a camera in your cell phone. Here you can see cells which acts like lenses, and here you can see cells which acts like photoreceptors. It's like matrix in your camera. And here you can see this light can go through these lenses, and then lens will understand, but not understand, of course, this light and react somehow. And our story is about the lens can use not only light signals but also electrical signals, chemical signals, and acoustic signals. I'm talking about ultrasonic sounds to communicate with each other, with another species. For example, some sort of plants can emit some odors to get attention of birds in case when some insects try to eat it, and these birds eat these insects, and plants will break again. Yeah, so we want to collect all this data, and using neural networks, we want to find if it's possible to find results. For example, we will show the same visual stimulus, and we will see the identical results of this output of neural networks. It's like we will find a pattern which will again tell, not tell, but tell that this plant can understand the same stimulus. Here, how it looks like in real life. Ocelli, it's the first layer of cells, look like this, and a lot of leaves was cutted. Yeah, suffering all plants. It's very interesting, but we are talking about a lot about ethic and moral when we communicate with plants, but in the same time we should cut them. Yeah, and we use microscope. Here you can see these small samples, and here you can see it, but it's a small paper with painted red star, and it looks like this is photo which we get from microscope, and here you can see these stars. It's maybe not the best chosen photo, but it was really like a star. Yeah, and our project raised questions about our communications, about our species interactions with plants, and as frankly I think that plants it's something more valuable that we used to think right now. I mean, normal people used to think, and it's just another creatures valuable like we are, like human on our planet Earth. Yeah, thank you for your attention. Maybe you have questions. Yeah. we don't have PowerPoint right now, but it will be a result of our future research. So now we are here on this stage, or now we know this facilities, they appear, they exist, and we can continue gathering data, plans, continue making our research.
Are there any proposed commercial use of this kind of photo sensing?
Yeah, maybe you will have a webcam instead of your webcam. You will have some plant connected to you.
Some plant cam.
Yeah, plant cam.
Okay, thank you.
You're welcome.
And well, you are listening to one of our current students, the current research that they are developing. They will continue there in 1 more year, so we are very excited to see what this generation will bring up. But now I also want to give the stage and the word to our graduates. Victoria.
Yes, thank you. Yeah, we actually studied together with Laura. Started together, but in different fields a little bit. But I also touched, of course, bioart in our courses, and I will tell about 2 projects that we created. But first, I will tell a little bit about my experience on the master program, because it was really exciting, and I think it was a great opportunity for me, because first of all, I met these beautiful women. We all studied together, and then we created art group that's called Bioroboty 019 or Bioroboty 019. First of all, it was because it was like easier to make projects, students' projects together for the classes. But then we thought that, well, we work together pretty good, and we like it. It's really fun. And yes, it started like students project, and right now we're still working together. We create art projects, we participate in different exhibitions, yeah, and live happy, happy life,
The
artistic life. Yes. And also, I want to say that master program gave me opportunity of the workspace, because I work here in art gallery as a coordinator. I help to create students, and not only students, exhibitions, and create parallel programs, and every project that we make here in master program and art gallery projects. Yeah, I'll tell you about 2 projects, as I said, that connected somehow to bioart. But firstly, I want to say that it's not so professionally made as, for example, Laura making, because she's a true bioartist. But we, our ideology is profanity, so it's really ironical. We get inspired by some science fields, and then create something really philosophical and ironical, sometimes really funny. And one of the projects we created while we were master students with Fizarum. We had a course with Natalia Fedorova, and she showed us this beautiful creature. Maybe Laura, can you say a couple words about it?
Yes, so Fizarum is usually known as a slime mold, and when it's growing, it looks like it is a fungi, but it's not. It's actually a protozoa it has a lot of nuclei together, and it's moving, it's growing, it's looking for food. We will show later a little bit more of it. But the most amazing thing of this organism is that it can have a little bit of memory of where he went, where it went, where was the food, and also create the shortest way to the food, to the other places where it's looking for. So it is an organism that we are studying the primitive intelligence of life.
Yeah, thank you. we get really excited about it because it looks like this on the hand, and it's visible, but we understood that it's one cell creature, but it's somehow smart. So yeah, we had a little workshop in classes, and then we created a project that called Fizarum Fortune Teller. We created fortune teller, modern fortune teller that connects biotechnology, like Fizarum, and like machine learning, computer science, and create modern fortune teller that can say what you're gonna be in the future. And of course, it's not true. It's ironical project, but people really believed in this in this one. Yeah, it was really fun, and actually, it started as a students project, but then Sveta, one of the member of our group, made this project as a graduation artwork, and it was like a year ago, right in September, the exhibition, and in this gallery we showed this here. And this project is a photo project, but we had a class with Ippolit Markelov. Really interesting classes in lab. We worked with everything that we had there. It's really interesting. And this glowing thing, it's actually bacteria with jellyfish protein that glows. So I don't know who scientists gave to bacteria this protein, so they started to glow. And Ippolit made inks with this bacteria. So we actually, this a square thing, it's petri dishes, and we actually print it with the real printer that we make, we have in every offices. We print it with bacteria, and we decided to print this text. It means that, well, it's about our, how you say province in English?
Regions?
Regions, yeah, thank you. So it's about regions, it's about difference from Central Russia and all the regions that we have, because a lot of people want to live in Moscow and St. Petersburg, and yeah, this project is about it, and it's called No Growth. And so yeah, we just had this workshop, we made this printing with bacteria and made photos, and actually you can buy these photos in Photo Department in St. Petersburg. So yeah, we try to make business from it. Yeah, that's it.
Yes, thank you, Vika. I think that one of the takeaway measures of this part, is all the different opportunities that you will have after the master by doing your own artworks, by earning money by making curatorial concepts, developing your own exhibitions. And again, like Vika said, that they are very ironical, and I think I also love that the fact that you could go deep into some field to understand it and still be able to make an artwork, let's say, against it, or finding the weak points. So that's why I'm super excited and happy every time I see a project of them. And when you finish the master, so there is a lot of possibilities. But I think now I would like to give the word to Lina Kiprushina. She will tell us how to study then in this master.
We have a question from YouTube.
Okay.
What did the masters give to you?
Okay, as I said, firstly, it gave to me art group. So we met in master program, and we're still working together. It's really cool. And I think the main thing that I get from these professionals that met all the curators and artists, because it gives a lot of opportunity. We met them in classes, and then they asked us to participate in the exhibitions. They gave feedback on our projects. Yeah, it's a really cool experience. And also, well, actually I didn't say that, but I'm from different fields. I'm optical engineer, so for me it was really convenient way to change the field of my interests. So I came from engineering to art, and this master program actually gives you this opportunity to, well, use all that experience, all experience you have. You shouldn't be the artist. You can be anyone you want, and yeah, you can become an artist even though you were an engineer or physics. Sveta, that I told you about, she's studied oil engineering. So yeah, this is pretty exciting.
Yes, Sveta now works as coordinator in a gallery, right? And also, like regarding your background, one of the wonderful artists that we have in our exhibition was from Vika, also working with optics, and now is in the Museum of Optics, so you can also visit there, one of her final projects.
Yeah, right. It's really exciting because I have my project in the museum.
Any more questions? Okay, then.
Okay. Hi everyone. My name is Lina Kiprishna. I'm a coordinator of the Art and Science Master's program, and I'll tell you about the admissions, how to get to the program. How can I?
The big button.
No.
So that means.
Yeah. Yeah. Okay. Thanks. So as the girl said, basically anyone can become a student at the master's who has a bachelor's or specialist diploma. You can be from any field, no matter what. It can be creative and engineering or a humanities field. And at the program you can learn how to be an art and science artist. You can become a curator. You can research art and science, so you can choose your own path at the program. This year we have 32 tuition-free bюджетных мест positions, and we have 15 fee-based positions. So I'll tell you about different options, how you can enter the program. You can see the tuition fee on the slide, and this year we also have one scholarship for an international student that covers all tuition fee throughout 2 years. So that's pretty cool. For that you can contact us. On the last slide you can see, you will see the contacts, and you can just write us an email and we'll let you know how you can process with that. So here are the admissions options. The first one is portfolio competition. We have two actually portfolio competitions. I'll tell you about the difference. We have online and we will have offline entrance exams this year finally, and also you can participate if you are the winner of the I'm Professional competition by Yandex. So the first option is Art and Science Portfolio competition. It has already started, so you can apply until June 15. The major characteristic of this competition is that, for example, you don't need to have any scientific publications or participate in some conferences, unlike in the next competition. Here you can You can submit a portfolio with works you already have or which you are planning to have when you join the program. So it can be some ideas for the projects you are going to work on at the program. Yeah, so your motivation and your skills are key here. Yeah, next option is IDMO University Portfolio Competition. So it's a contest that is common for the whole University of IDMO. That's why it has some standard points. For example, your exam grade should be no longer than 4.3. And for example, at the previous competition, your exam grade doesn't matter. And also here you need to have a proof of at least one report, one publication, or for example, some conference publication can also work. So this kind of contest is more suitable for those who have some maybe scientific background and not an artistic one. Also, you can apply for it already now. You cannot apply for both competitions. You need to choose one of them. Then we have entrance exams. As I said, they will be online and offline. They will start in May, and the last exam this year will be on August 10. So there will be a lot of dates. Soon they'll be published on the website of IDMO. You can just pick one of them and take the exam. So the exam takes place in English. Basically, you get a ticket. You write answers to 2 exam questions there, and then you have an interview with the curators of the program. We are going to have pretty soon, next slide, Art and Science Master's course. It's a course to help you get prepared for the exams. It has it takes 7 weeks, and apart from every week session, there will also be some additional lectures that are devoted to some topics which will be useful for you if you take this option. And you can apply for it already now. You can go through this QR code and fill out the application. And last year, about 40% of all the students we had took this course and enrolled in the program. And the last option is unprofessional competition. It's for those who take part in this Yandex competition, and to get to the program without any exams, you need to be the winner of the design category. So already in May the winners will be announced, so if you participated, this can probably work for you. You can take one of the portfolio competitions, and for example, if you don't win, you can still take the exam and enroll in the program through this way. Yes, if you have any questions to any of us, please don't hesitate. Yeah, and here are our contacts. You can write us anywhere and ask questions.
Thank you for your presentation. So you said that for the Art and Science portfolio competition, you don't have to have any middle average grade from your major, and you don't have to have those publications.
Yeah.
Okay. Yeah. Thank you.
So you only need to have a portfolio with a motivation letter. That's the whole application.
And it's like a big variety of, it shouldn't be your portfolio in art and science. Maybe you have been drawing something, you have different experience, something that you could bring to this master.
Yeah, even if you don't have like art projects, even traditional ones. It can be maybe some scientific projects or some startups, like anything you worked on.
Actually, I get the masters from portfolio. Sorry, from portfolio, and I had scientific portfolio, so I put there my article about optics and my engineering experience. Yeah, and in motivational letter I explained why I want to study art. Yeah, and that worked, I guess.
Do you have any other questions? Maybe online? Okay. Well, if you are ready to dream, learn, and create, I think we will be happy to see you here. If there is no more questions for online, we will stop the online transmission. But now, for those present here, I invite you to take part of a really small workshop working with slime molds. Vika explained about this Fisarum, and if you are up to, we can go and you will see it